So for our next talk, as promised before, we are going to be leaving the gut, at least for a little bit, and going up to the brain. And of course, understanding special nutritional requirements as a result related to the brain is very important because of the issues of the blood-brain barrier. So thank you very much. We have Martha Field from Cornell University to be talking about that.
I'd like to start by thanking the organizers for the invitation. It's been a real treat so far, and I'm really looking forward to the rest of the workshop. So today I'm going to talk to you about blood-brain barrier dysfunction and resulting nutrient deficiencies. So here are my disclosures, and I'm just going to orient you to my talk by showing you a slide that Dr. Stover already showed, but I'm going to be talking about several disease-related factors, including inflammation, genetic predisposition, autoimmunity, and mitochondrial dysfunction, and how, in my talk, that affects the blood-brain barrier, and then impact on really a tissue-specific or a brain-specific nutrient deficiency.
Goodbye.
And I'm going to talk a little bit about tissue-specific biomarkers. And I'm going to focus today on cerebral folate deficiency. So I'm just going to keep going here. Okay. So just a quick review on the CSF and brain nutrition. And so the cerebral spinal fluid really surrounds the brain. It serves as a nourishing fluid for the brain, and it serves to remove waste products. And so blood-brain barrier function is really critical for maintaining brain nutritional status. And there are two components of the blood-brain barrier. There's an endothelial component and an epithelial component. And although the players are different in terms of the proteins that transport nutrients at each of these respective barriers, and the proteins that form the tight junctions are different, the playbook is the same. In many cases, these barriers serve to transport and, you'll see, to concentrate nutrients. Vitamin C is concentrated fourfold in the CSF relative to the serum. Many of the B vitamins are concentrated, some as much as 50-fold. And again, I'm going to be focusing on folate, which is concentrated fourfold in the CSF relative to the serum. And there are many different mechanisms by which these barriers concentrate nutrients. And so it's well known that blood-brain barrier function declines as a result of disease. It's perhaps most well characterized with respect to Alzheimer's disease, but blood-brain barrier dysfunction is also known to result from multiple sclerosis and from HIV infection. It's also well known that there is a natural decline in blood-brain barrier function with age and as a result of inflammation. And there are really several factors by which this happens. In some cases, there is a loss of transport protein expression, and in other cases, the tight junctions physically erode. And so when you're trying to concentrate a nutrient from the blood to the CSF, you're really depending on that nutrient not just leaking right back out. So you need those tight junctions. Oops. And so I'm going to be talking about cerebral folate deficiency, and I realize this is a very busy slide, but I want to use it just to illustrate a few points. So folate in the cell, shown as THF, the purpose of folate in the cell is to carry and chemically activate 1-carbon units for biosynthetic reactions. And different forms of folate can be interconverted by a series of metabolic enzymes. But the bottom line is that you need these biosynthetic reaction. You need folate. You need folate for synthesis of purines. That's the A and G base in DNA. You need folate for synthesis of thymidylate, the T base in DNA, so 3 of the 4 DNA bases. And you also need folate for the remethylation of homocysteine to methionine. Now, methionine is a precursor of S-adenosine methionine, and that is a methyl group donor. And so you need S-adenosine methionine for methylation of DNA, histones, and in synthesis of neurotransmitters. And so that's why you need folate. I also want to point out how folate gets into the cell. There are 3 known transporters there's two transporters and one membrane receptor. I'm not going to really talk about this transporter because that's responsible for intestinal uptake of folate. But most other cells express either the reduced folate carrier, also a transporter, or the folate receptor. And so let's dig into those two transporters a little bit more. And so the reduced folate carrier, again, is a transmembrane protein. It has a low affinity for folates, but a high capacity for transport. Its expression is fairly ubiquitous. Its expression is regulated by folate. In the mouse, mouse knockout is catastrophic, and these embryos die prior to neural tube closure. This is also the transporter that transports antifolates or antifolate chemotherapeutic agents. The folate receptor is different. So this is a GPI-anchored membrane receptor. Unlike the reduced folate carrier, it has a very high affinity for folate, but that then gives it a low capacity for transport. It has an exceptionally high affinity for folic acid, and we'll talk about what that means at the blood-brain barrier in a minute. And again, its expression is not ubiquitous. It's really in a limited subset of tissues. And I'm most interested in today's presentation about its expression at the blood-brain barrier. Its expression is also regulated by folate, and the mouse knockout model develops neural tube defects. But let's look in a little more detail about folate transport across the blood-brain barrier. So remember, there are two blood-brain barriers, essentially. Folate is transported at the blood-CSF barrier, or the choroid plexus. And so, as I said, folate levels in the CSF are about fourfold higher than they are in the blood or the serum. And this transport is mediated by the folate receptor. So folate binds on one end of this epithelial cell. The receptor is endocytosed, and it transports folate to the CSF. And so this transport is in the choroid plexus. Folate transport is absolutely dependent on the folate receptor. And because of that endocytosis, it's ATP dependent. And so the bottom line is, for transport of folate into the brain, you need the folate receptor and you need energy. And we also know that because of this difference in folate concentration in the CSF relative to the blood, the CSF or CSF folate levels are really reporting on brain folate status independent of indicators of whole body status. And so what do we know about inborn errors of metabolism that lead to cerebral folate deficiency? And so there are, in fact, a number of studies demonstrating that individuals with inborn errors, again, these are genetic defects in either the folate receptor or genetic disorders that cause mitochondrial DNA depletion. So either a mutation in the receptor or a loss of energy that then causes cerebral folate deficiency. And you can see the normal range should be, let's say, around 100 nanomolar. And in these cases, CSF folate is very low, and in some cases not detectable. The intervention, you see in every case here, is very high dose folinic acid. And folinic acid is different than folic acid. And what you see is that treatment with this reduced form of folate, folinic acid, in fact, improves CSF folate levels and results in clinical improvements. And so what we can take from the inborn errors of metabolism literature is that in treatment of cerebral folate deficiency, folate form matters. And so folic acid is different than folate within the cell. Okay, and I'm going to explain a little bit of that. Folic acid is a synthetic form of folate. It's highly bioavailable and it's chemically stable. It's reduced to a natural cofactor form once it's inside the cell. Studies show us that, however, folic acid is not as effective at reduced forms of folate, like folinic acid or like methylfolate. Methylfolate is the B vitamin form. That's what you actually find in fruits and vegetables, but it's not very chemically stable. It is much better at improving CSF folate levels. And there are probably many reasons why folic acid isn't effective. These are just a few of them. So it doesn't do a very good job of crossing the blood-brain barrier. And there's some evidence that because it binds that receptor so tightly, it may actually impair transport of methylfolate, the reduced folate, which is what is found in your blood, across the blood-brain barrier. And in fact, at such very high doses that are shown to be effective, folic acid can be associated with some toxicity. And so several authoritative bodies have recognized the relationship between impairments in whole body folate status and brain disease, or more specifically, depression. The Institute of Medicine in 1998, the World Health Organization in 2008, and the American Psychiatric Association in 2010, and they commented that folate is a low-risk intervention with general health benefits. How does this association between whole body folate status parallel with what we see with the inborn errors of metabolism and a tissue-specific nutrient deficiency? So we talked about the mechanisms whereby genetic inborn errors lead to cerebral folate deficiency. In adults, although the causes are different, the end result is the same. And so blood-brain barrier dysfunction in age-related chronic disease is associated with folate receptor autoimmunity, so antibodies that bind the receptor and prevent folate from binding. It's also associated with pharmaceutical use. Certain pharmaceuticals, including drugs of abuse, are known to impair blood-brain barrier function. And again, this is an energy-requiring process. Mitochondrial dysfunction is also a component, and that's associated with age and with chronic disease. There's also a genetic component to the link between cerebral folate deficiency, or between low folate status and depression. So there's a common genetic variant that affects both functioning of folate-mediated 1-carb metabolism and folate status. It's a single nucleotide change in the MTHFR gene that makes a single amino acid change that destabilizes the protein. That effectively causes a low folate status. NTD risk is doubled for carriers of the TT genotype, and it also is associated with increased risk of miscarriage. Interestingly, in folate-sufficient adults, however, the TT genotype is protective against colon cancer. And so because of this low folate status, the MTHFR T allele has been associated with increased risk of developing both depression and schizophrenia. And it's known that up to 70% of patients with either depression or schizophrenia have some form of the polymorphism, so either one or two T alleles. And it's worth noting that the prevalence varies by population, but in most healthy populations, it's less than or equal to 40%. So major depressive disorder is projected to be the second leading global disease burden by 2020. And standard pharmacological treatments, so SSRIs, yield a response to medication in less than half of all patients. The etiology of major depressive disorder is poorly understood, but it's likely to involve a complex interplay of sensitivities to stressors and alterations in metabolic pathways. There is some evidence using peripheral biomarkers that blood-brain barrier integrity is compromised in depressed patients. And so we talked a little bit about what the observational studies show in terms of genetic polymorphisms and risks for depression. And so now let's ask what the trials show. And the trials are decidedly mixed, but I think still informative. And so one trial used folic acid for 12 weeks in depressed patients and found that it was ineffective for reducing patient-reported or clinician-rated severity of depression symptomology. This tells us, just like in the inborn errors of cerebral folate that cause cerebral folate deficiency, that folate form matters. Folic acid is good at a lot of things, but it's not good at improving cerebral folate deficiency. What about the reduced forms of folate? So there are two trials that showed that 15 to 30 milligrams per day of methylfolate, so this is reduced folate, resulted in significant declines in depression scores, or again, a reduction in severity from one category, so from severe to moderate. It was also clear that lower doses did not result in symptom improvement, suggesting that dose matters or that there's a dose-response relationship. And in fact, 30% responded in this trial to methylfolate plus an SSRI, compared to only 14% having symptom improvement if they were taking the placebo and an SSRI. And so clearly, here is an issue of responders versus non-responders. And I do want to point out the dose. These doses are very different than requirements in healthy individuals. And they're different, it appears to be, by more than an order of magnitude. And so what I want you to take away is that although the origins of cerebral folate deficiency are very different when we're talking about inborn errors of metabolism compared to adult onset cerebral folate deficiency, the treatments are essentially the same. The treatments, I should say, that result in clinical improvement are essentially the same. And so what have we learned from cerebral folate deficiency? So I hope that I've convinced you that brain-specific folate deficiency can occur in the absence of whole body deficiency, in inborn errors of metabolism, and in an adult disease. Although it's important to point out that whole body deficiency could exacerbate the condition. If you're pumping folate from the serum to the CSF and serum levels lower, that then exacerbates the condition. I have also demonstrated that blood-brain barrier folate transport defects can be overcome with very high levels of folate to overcome the physical and energetic barrier, and that folate form or composition matters for efficacy. Reduced folates are efficacious, folic acid is not. And so that leaves us with a model whereby folate administration in disease meets this increased requirement in disease by overcoming the blood-brain barrier dysfunction, to restore brain folate status, to restore biochemical function in the brain, ultimately leading to clinical improvement. And so just to quickly summarize some of the questions we were asked to address. So first, what is a biological mechanism? And in this case, it's blood-brain barrier breakdown. And we were able to draw on a relatively rich inborn error of metabolism literature to understand these mechanisms. The second was, is there evidence for nutritional requirement that's different than in a healthy population? Absolutely. Is the disease state responsive to the intervention? And I would say yes, but, and that's where we get into some of the complexities. There are clearly responders and non-responders. And I think understanding these relationships really requires an understanding of the biomarkers and rigorous, well-designed controlled trials so we can begin to understand these relationships. There does seem to be evidence of dose response, and then what are the research gaps? And I'm again going to return to this slide and just remind you that when we're talking about nutrient needs and disease, we're often talking, and this is a really good example of it, of a tissue-specific nutritional requirement. And again, we need biomarkers to help us understand how to go from the biology to the function to ultimately tissue regeneration. And so what are the information gaps? So it turns out, again, as I mentioned earlier, there are several nutrients, not just folate is important, there are several nutrients that are concentrated within the brain, and we don't really have a good understanding of how these nutrients are affected by disruptions in blood-brain barrier activity. We also don't understand whether repairing the barrier can reverse the nutritional deficiency and or whether restoring brain nutrient levels can augment blood-brain barrier repair. And again, I think perhaps one of the most pressing gaps is that we really need good biomarkers, we really need robust and rigorous trials, and we need appropriate model systems for this. And so I would just like to quickly thank Patrick Stover, Amanda McFarland, and Jane Durga for thoughtful discussions.